Okay, so it's a great pleasure to have Professor Angus Murphy here from the University of Maryland. He's the head of the Department of Plant Sciences, and he has been spending the last 20 years understanding the role of the plant hormone auxin, and in particular how it's transported around the plants by a group of special proteins known as ABC transporters. And Angus's work has been instrumental in our understanding of how auxin controls plant development, and he's taken the fundamental biochemistry that he's elucidated in the lab and translated that into improvements in food yield and crop yield in the field, and then has taken that knowledge and translated it back into our understanding of fundamental biochemistry in the lab.
So today I gave a lecture, and that lecture was describing the role of auxin transport in both model and crop species. And the emphasis was on the translation of what we've learned from work in model species, particularly to maize and to sorghum, but also to some other crop species. The other part of the discussion was how what we've learned in crop species translates back into what we know in model plants, and how that type of exchange of information is essential to how plant biology is going to proceed in, well, the next decades, let's say. It's important because the topics that I was discussing are very relevant to crop responses to stress, and very particularly to water shortage and to production under limiting conditions of nutrients and sunlight. These are issues because increasingly we are engaging in what's called precision agriculture, wherein we have to deliver exactly what is needed to the plant at a specific time for optimal growth. So the work that I was describing is rooted in the transporters that mediate polar movement of the phytohormone auxin, which is important not only for fundamental development in plants, but also for the plasticity of that development under environmental inputs. It also extended to trying to understand how a family of proteins that mediate this transport may have other functionalities, and emphasize the importance of integrating structural information about the proteins with assessments based on gene assembly and the transcription or expression of those genes. So the fundamental message was that we do know something more in the case of these transporters called ATP binding cassette transporters, or ABCB transporters. They're a subclass of this type of proteins, and how they function in exclusion of compounds, particularly auxin, from the plasma membrane. And although it seems trivial, this function is an important part of the overall long-distance transport pathway. Unfortunately, what my talk also emphasized is how little we know about the different components of this process. In other words, mechanistically, we have some idea of how to break out the efflux of the compound from the cell. And we know that there's another component that has to do with exclusion, and another one that has to do with a kind of leakiness to small ions. But we still haven't actually sorted out which component is most important under a particular circumstance. So the discussion went on to consider what might be the most profitable way forward, and how much of that should be done in model systems and how much should be done in crop plants. In the long run, the final estimation was that we have to collaborate in an interdisciplinary way to accomplish our ultimate goal.